Good morning, everyone. Hello, we'll start today's webinar on Wednesday, the 13th of December. Thanks for joining us. Today we're particularly talking about from the ocean to the plate, impacts and hot tips for seafood. My name's Dr. Emily Osier, and I'm one of your co-hosts along with Professor Greta Pessel. And we've organised this series of webinars at the start of what's going to be a long, hot marine summer in Tasmania. And we're really looking forward to your comments and communication and questions via the chat. And we'll have a Q&A session at the end. Thanks for joining us today. And I'm going to be your host, but we'll have the session chaired by Dr. Katie Creswell. And Dr. Katie is an ecologist and fisheries modeller, and she and her team down at IMAS work on addressing problems in fisheries and ecology. But before we get going, I wanted to acknowledge that those of us joining from the IMAS campuses are joining from the lands of the Muwinina people. And the Muwinina people, as the traditional owners and custodians of these lands, are not with us today as a result of colonisation. But at the same time, we acknowledge the continuing knowledge and custodianship of the Tasmanian Aboriginal community, and particularly given what we're talking about today, their knowledge of living through times of extraordinary disruption and change. So the purpose of the webinar series that we're operating here and now is to share information about the current marine heatwave conditions that we're experiencing in Tasmania particularly, and that's part of IMAS and UTAS's commitment to place. We're also here to talk about the kinds of impacts that we're expecting on Tasmanian marine ecosystems and marine sectors and communities, on our blue community, if you like, and also to share information and actions that are underway to give us some sources of hope and actions to take. So I'm now going to hand over to Dr. Katie Creswell to chair the session, but we really look to see these events as interactive. Please give us, as I mentioned, use the chat to share information, and also be aware that the slides and presentations and recordings are going to be available on the CMS website in the days to come. So thanks for joining us. I'm going to hand over to your chair, Dr. Katie Creswell. Thanks, Katie.
Thanks, Em. Yes, I'll be chairing today. I'm a research fellow at IMAS, as Em said, in wild fisheries modelling, with a special interest in range extending fisheries and also the impact of climate change on fisheries. So this is going to be a really interesting session. The webinar topic today is From the Ocean to the Plate, Impacts and Hot Tips for Seafood. We've got 4 presenters today with about a short presentation each, about 6 minutes. And we're going to start off with Dr. Jaime McAllister from IMAS, looking at the heat stress impacts on abalone. Thanks, Jaime.
Thanks, Katie. Yep, my name's Jaime McAllister. I'm a researcher down at IMAS Taroona, work in the dive fisheries section, and my primary or my main focus is on the abalone fishery in Tasmania, particularly the commercial fishery. So yeah, today's presentation is just a bit of an overview of what we're doing and what we have been doing in relation to some of the physiology and heat-related stress events on abalone that we've witnessed over the years and going forward. So without further ado, so temperature and abalone physiology, this is just a basic sort of background. Like most marine organisms, abalone are cold-blooded and therefore influenced by their surrounding environment. However, unlike fish, abalone are relatively sessile, as you would expect. So therefore, they need to sort of tolerate or adapt to the prevailing environmental conditions. They simply can't just up stumps and move like some marine fish that we've seen over the years. So subsequently, don't read too much into the detail of these figures, but it's just sort of demonstrating that subsequently this will affect the physiological processes. So as temperature increases, physiological processes such as reproduction and growth will be influenced as the metabolic rate increases. So it's a bit like us, as temperature increases, our respiratory rate increases as well. So that comes at a higher energetic cost, and that can affect the performance of various metabolic processes such as reproduction and growth. Obviously, this leads to increasing stress as energy reserves are redirected from key processes such as growth and reproduction just in order to survive a warming event. Generally, for black lip abalone, we consider the optimal temperature for growth and reproduction as around 18 degrees centigrade. And then if we think of what the critical limit is, so the point at which they potentially would start to sort of see mortality event, is around 26 degrees. So the thermal optimum, and generally above that, that's when we start to expect mortality events occurring. So now mortality events are fairly obvious, as we witnessed back in 2016 when Tasmania experienced a fairly prolonged marine heatwave off the east coast. It was estimated that we lost somewhere between 5 to 10% of the stock of abalone along Tasmania's east coast. Interestingly, though, that occurred about 7 degrees below their thermal maximum, which suggests that there is some localized adaptation occurring in populations along Tasmania's east coast. However, what was probably less obvious was some of the sublethal effects of exceeding those thermal limits on abalone physiology, and in particular, the effect on reproduction and recruitment into our fishery. One of the key questions we have is there any suppression of reproductive activity during these warming events as sort of abalone redirect their energy reserves towards just survival? Are they redirecting it away from reproductive activity, and thus we're seeing suppression in reproduction and recruitment? Now, and why is that less obvious? Well, it sort of takes 6 to 7 years before we start to see recruitment in the wild as abalone emerge from cryptic habitat. So it's a bit of a lag response, and in many cases that's just too late to make effective or sustainable fishery management decisions. So that's a bit of a lag of 6 to 7 years before we can sort of make key decisions. And unfortunately, this scenario appears to be playing out right now in Tasmania. A lot of our fishery independent surveys at the moment, we're seeing very little recovery in the East Coast population and seeing very little signs of recruitment in our fishery in the years preceding the 2016 marine heatwave. And that sort of makes sense. You know, if we think 6 to 7 years back when the marine heatwave event hit, this year being 2023, it suggests that perhaps there was some suppressed reproductive activity that occurred as a result of that 2016 marine heatwave event. Fortunately, we've had some advancements in various technologies, and there's this omics technologies, and essentially what that is just looking at a higher level molecular understanding of the structure and function of organisms. And basically we're looking at, you know, how does different environmental stresses, toxins, heat obviously, and then even in the aquaculture setting, how do those affect the organisms through time, so from juvenile stages right through to adult. And we can use these omics technologies to better understand some of the, well, essentially they're tools that can use how various environmental stresses affect, sort of, if you think of an abalone, it has all these metabolic pathways inside, and one of them being obviously the key one, reproduction and recruitment. How do these environmental stresses affect those abalone at that higher level that we can then use as a forecasting tool to see what happens 6 to 7 years down the track potentially? So what have we been doing? Well, since 2020, we've been examining the spatial and temporal physiology of abalone along Tasmania's east coast. I had 6 sites in 2020 that I've been collecting animals from and understanding the physiology of those animals using those omics technologies. Yes, we're seeing spatial and temporal variations in those, still writing up those results. But in wake of the marine heatwave, we've continued to collect samples from 3 sites to get that sort of spatial distribution. And we're looking at the pre, post, and during the marine heatwave events. So I've basically been collecting the last 3 months, and obviously we'll continue that through probably until about April. When it's expected to subside. And ultimately, the aim of this research is to sort of understand how increasing water temperature is potentially affecting those reproductive recruitment events, and obviously some growth in there as well, but really just to provide that forecast to potential recruitment or productivity gaps in our fisheries, and just allow us to set more I guess, conservative management strategies well in advance. And I think that's it for me.
Awesome. Thanks, Jamie. We've already got some questions in the chat. You could have a look. I will answer them after. And next up, we've got Associate Professor Quinn Fitzgibbon.
Hi, everyone. My name's Quinn. I'm a lobster physiologist in IMAS. I work closely with aquaculture and also the fishery, particularly through the post-harvest sector. So my story starts back in 2016-17. What happened is that there was a few facilities, export facilities, lobster export facilities in Tasmania that experienced some really high levels of mortality, really severe mortality events. Initially, these events were thought to be localized, and potentially the theory was impacted by toxicity, potentially related to some localized mining activity. However, as I got called in to investigate this, and as I started looking into this, it became very apparent that soon other severe mortality events were starting to pop up, and different companies across the entire sector. The sector got really concerned at that stage. They were concerned that there was a new industry, a new disease that was leading to these mortalities. So we were called in to do quite a large research program to understand the mortality and particularly look for a disease. And we did a whole heap of work on the epidemiology, physiology, and pathology of the lobsters. To cut a long story short, our studies did find pathogens, but it did not support a systematic crustacean pathogen associated with mortalities across the entire sector. However, it was really clear that there was an increased sensitivity of lobsters in these seasons. And I suppose our hypothesis at that stage that there was a reduced holding capacity as a result of high physiological stress, reduced immune function, which was leading to overwhelming infections with secondary bacteria or loss of physiological maintenance, leading to death. At that time, there wasn't a particularly strong awareness of the heatwave event that was occurring, but we certainly started to draw the links to what was happening, because these mortalities really corresponded with an unprecedented marine heatwave in Tasmania that you've already heard about. And particularly, I was concerned about the impacts of environmental stress, but also impacts on molting cycles. So not only were the animals more stressed, but they were also molting out of season, and molting, both pre and post molt lobsters perform poorly in the post-harvest sector. Very interestingly, New Zealand over the last couple of seasons has had a similar heat wave, and they've had very, very similar problems with the quality of the lobsters and mortality in post-harvest. And so our investigations we saw there was a concern, but our investigations also revealed that there was considerable differences in management processes between companies and apparent level of mortality. And so this really suggested that there was an impact of post-harvest practices. What may have worked in the past wasn't particularly working well during this hot seasons. And we particularly identified different areas of the post-harvest sector that were potentially suboptimal. And so our project actually moved to try and improve industry practices. And so we did a big analysis of how the lobsters were managed. And then we conducted a whole series of work to try and improve practices, including water testing quality approaches. We created a best practice guide, which had recommendations for all the aspects of post-harvest handling. And we conducted industry best practice workshops across the entire sector. And I'm very pleased to say that the industry was very open to this and adopted and improved practices, which was great. However, what is clear is that the performance of our lobsters is really dependent on the sum of the experiences through the entire chain of custody from capture to market. And our previous work really looked at the factories. But it was also clear that the fishing industry has an important part to play. And so subsequently, we've been working with the fishing industry to do a similar sort of analysis of practices and processes. What's really clear is that we have an upcoming heat wave, and so good practice is more important than ever, particularly this season. And so we have initiated a heatwave critical response pack where we're highlighting key factors for the industry to take care of this season. In particularly things like carrying capacities, making sure they have the appropriate water flow, recording water quality in their wells, and insulation of aeration as some key ones that we're trying to focus on. However, again, there's a lot more to the activities, and so we've conducted an analysis across the industry and produced a best practice guide, which covers all aspects from collecting the stock to sorting, grading, holding conditions, landing and offloading. We're extending this further with wheelhouse guides, and right now, as a priority, I'm conducting, again, industry workshops across the sector. Just last week, I did Port Lincoln, Robe and Port Macdonald, and we'll be doing Tasmanian ports in the very near future. So that's all I've got, Katie. Thank you.
That's great. Excellent. Thanks, Quinn. We're just going to go straight to the next one. Dr. Daniel Pountney on the aquaculture, feed and husbandry.
Okay, good morning. So my name is Daniel Pountney. I'm a lecturer and researcher here at IMAS in the sustainable aquaculture side of things. And I teach into nutrition, physiology, and production themes. So my background is in commercial aquaculture and commercial aquaculture feed development and management here and also overseas. So when we talk about aquaculture, it's really, the purpose of it is to grow nutritious food in a responsible and sustainable manner. So I will just briefly discuss some of the temperature-related impacts that affect fish performance. So you've heard already today from Jaime, but temperature is one of the most important environmental factors influencing the biology of ectotherms. So ectotherms represent about 99% of all fish. So basically, if it's hot outside, it's going to be hot inside. They don't have the ability to regulate or maintain their body temperature. So it's regulated by the temperature of the water that they live in. And fish have a thermal tolerance, and this will differ between species and also between geographic locations. And what I mean by this, in this figure here on our right, we can see we've got this red line, and on the left, we've got low temperature moving towards the right to high temperature. So fish will have a temperature optimum where they perform at their best. So performance might be in terms of growth performance, and that's what we're really interested in when talking about aquaculture, high performance, which means usually high efficiency. So if we go to the left of the curve, we have lower temperatures and the metabolic rate or the metabolism will drop off and they will be growing at a slower rate. And if we go beyond our temperature optimum, it sort of declines quite quickly, okay? And this is where the species might be in their heat stress area, where they will reduce their growth performance and they'll reduce feed intake. And this is an area that is problematic during warming waters because it reduces the efficiency of aquaculture production. So when fish species might be impeding on their thermal tolerance, let's say onset of heat stress, say if we use the Atlantic salmon industry in Tasmania, in the key industry, we might think that the temperature optimum could be between 12 and say 18 degrees Celsius. And beyond that, there will be a decline in feed intake. So feed intake will have very similar curve to growth performance. So feed intake drives growth performance. So when they're not feeding, they're not growing. And when it gets to a temperature sort of around 20 degrees, they will actually be losing weight. They will be using those energy reserves to maintain like homeostasis, for instance, maintaining balance within the body. So when we have reduced feed intake, we also have reduced growth performance, which is again not great for production. And what happens is we see a decrease in the efficiency overall. So the efficiency will be maybe FCR, we see reduction in food conversion ratio, or actually it's increased, so it's a negative. It might be that the fish in, fish out ratio, so the amount of marine products consumed by the fish to harvest is also increased as well. And ultimately there is a decline in the health status. So the health status could be that the fish are utilizing those essential nutrients that as a consumer of fish, we really are looking to eat. So let's say essential fatty acids and vitamins and minerals, which are really important in fish for consumers. Okay, so temperature is one of the things we're interested in, but also oxygen is the other. So oxygen, again, essential for life. That shouldn't be new information. It's somewhat limiting factor for metabolism. It's essential for biological, biochemical processes within the body, and high metabolic rates require more oxygen, which can be really problematic when the environment is low in oxygen. When we look at this figure here, we can see our oxygen concentration on the left, on the y-axis, in milligrams per litre, and temperature down the bottom. And we've got two scenarios here, one for fresh water and one for seawater. Again, if we're considering Atlantic salmon grown in salt water, we can see that there is less oxygen available due to salt water has a lower carrying capacity for oxygen than fresh water. So with increasing water temperatures, they have an enhanced metabolism. They require more oxygen, but there's less oxygen in the water. And that's really problematic during these hot periods, but also when there's a very slack tides where there's not a lot of water movement. So, you know, fish or aquatic animals are held within a contained environment, whether that's a cage, a tank, or a pond. They can't move away from their containment. So they rely on water being rejuvenated or passing through a cage, for instance. So during thermal stress, okay, there's uncontrolled dissolved oxygen. It's very difficult to add oxygen in many culture environments, in particular sea cages. If it was pond aquaculture, you can add aeration using aerators, or in a RAS, you can actually control oxygen on land. But in sea cage aquaculture, you have limited ways or limited number of tools to use. So during these low DO and also hypoxic events, it causes physiological stress on the animal. Again, they're going to reduce their feed intake. And feed intake is really important because their feed source is a complete nutritional package. Most species don't obtain any nutrition from the environment. It's all within the feed that they're provided in an aquaculture environment. And when you have that reduction in feed, again, you're going to have a decrease in growth performance and also a decrease in health status. and that could bring on onset of potential diseases. So they're more at risk to getting diseases. Ultimately, hot water impedes the metabolism and the oxygen availability, and it reduces the performance. And performance is really critical when we're talking about large-scale aquaculture. So what are we doing here at IMAS? So some of the research focuses include understanding the thermal tolerance and the growth performance of fish, and that's across multiple species. Understanding the thermal tolerance will give us some tools to combat or to enhance growth performance. Looking at thermal and hypoxic stress on gut physiology and morphology, and how the gut changes during these warming events, because the gut is a really important organ. That's where the digestion happens, and it's also really important for immunity as well. Also looking at the role that genetics play in changing environmental conditions that might drive tolerance and performance. And this is really important for expanding on selective breeding programs. Also on the nutritional side, determining these essential nutrient requirements and also the nutrient digestibilities during these periods of elevated water temperature and or low dissolved oxygen events. So ensuring that we're able to boost the fish before a heat wave comes along. Also looking at advanced monitoring technology to monitor jellyfish threats and fish condition in high energy offshore environments. Being offshore, it's not close to shore, so we don't have access all the time due to poor weather. It limits access to these sorts of facilities. So looking at a technology that we can utilize to monitor the health and the fish condition. So by looking at these different themes, it's going to help scaffold aquaculture industries in Tasmania, but also across Australia to support their performance during these warming periods. Thank you very much.
Thank you so much, Daniel. That's awesome. Okay, and next we have Dr. Andreas Seger on tips for harvest and supply of food-safe seafood.
Excellent. So I'm a research fellow down at IMAS. I work mainly in seafood safety with a focus on marine biotoxins. And I also work for SafeFood, which is the Australian Seafood Safety and Market Access Program. So today we're going to have a quick look at some of the things we can do to make sure that we eat the best quality seafood possible. And one of the key things to be aware of is, of course, what are our risks associated with seafood. So we got two main categories. We've got chemical contaminants such as marine biotoxins and heavy metals, and also microbiological hazards, so bacteria, viruses, and parasites. And they're all through the natural environment, so they can enter seafood at the time of harvest, or there can be secondary contamination during processing. So norovirus, for example, is a pretty notorious example here. Once they enter the seafood, if the conditions are right, these things can multiply, and then if they increase to levels that are of risk to us, of course, we may get sick if we consume that seafood. Yesterday, Gustav Hellegraf did a great job talking about phytoplankton blooms and what we might expect with in the heat waves coming through in terms of growth. And of course, the consensus is that it's not as simple as increased growth with increased temperatures. There's lots of other factors that play a role here, but it's important to note that some of these phytoplankton species can produce paralytic or diuretic shellfish toxins that can accumulate in seafood up the trophic levels and, of course, make us sick if we consume that seafood that's contaminated. So now that we looked at all the bad things that could potentially happen, it's worth noting that all the seafood that's consumed around Australia daily, there's very few cases of seafood-related illness. And that's mainly because we follow some very key protocols on how to make sure we get the best quality seafood. And the number one thing we can do is to keep things cool, just to limit the bacterial growth and bring the temperature down as quickly as possible. And the catchphrase here is keep it nice, put it on ice. So if you're out in your tinny and fishing, don't let your fish flop at the bottom of the boat and keep fishing, put it straight on ice. Same thing if you go to the shops, bring a cold bag, whatever you purchase, put it straight into the cold bag and take it straight home. Don't let it fester in the back of a hot car. Take it straight home, and then when you're processing it, practice good hygiene. So, a key thing here is don't prepare food for others when you're feeling sick, especially if you have gastrointestinal symptoms. For bacteria, cooking generally dramatically reduces the risk, but other things like the chemical contaminants aren't impacted by cooking, and we'll talk about those a bit more in a second. If you go out harvest recreational shellfish, it's important to know that water quality, you can essentially think of it as the equivalent of shellfish quality. So don't go collecting shellfish near marinas or stormwater outfalls or areas with septic tanks, especially after heavy rain, because it just flushes everything from the land into the sea. And that includes things that are transmitted through feces. So again, norovirus is a good example here. In terms of commercial seafood and quality control, it's very important to know that seafood in shops and restaurants is safe to eat. And that's mainly because there's very stringent protocols that these establishments follow, and that includes very strict harvest procedures. So when to harvest, when is it safe to harvest, then processing, lots of cleaning protocols, making sure there's no secondary contamination. And of course, the most important one, keeping the cold chain going all the way from harvest to when it gets to you, the consumer. And then it really becomes your responsibility to make sure that you keep that cold chain going. In terms of biotoxins, we have very well established phytoplankton and biotoxin monitoring programs throughout Australia. This map here gives you an idea of some of the sampling locations along the east coast of Tasmania. And if biotoxins are detected in any of these higher risk species, then commercial harvest immediately ceases, and there's also public health warnings available on the health department website. So before you go out harvesting any of these species, it's worthwhile just jumping on that website, having a quick look, see if there's any biotoxin activity in the area that you're intending to harvest. Another thing that we might see with sort of increasing temperatures is the southward trend of some of those highly sought after fish species, especially for anglers. Swordfish is a prime example here. And one of the risks associated with these higher trophic species is mercury. And the advice here is to limit the consumption of game fish to 1 serve per week. And for pregnant people, that's even less. That's about 1 serve per fortnight is the general advice here. So in summary, the most important thing is keep it nice, put it on ice. Make sure you keep that cold chain going all the way from harvest to when you consume the seafood. And if you have any further questions that we can't answer on the time frame here, there's lots of information on the Safe Fish websites. We've got lots of fact sheets and technical reports of some of the work that we're doing. Also webinars on Vibrios, for example. So if you're interested, jump on the website and have a quick look. And that's all for me for today. So thank you for listening.
Great. Thanks, Andreas. Thanks, all presenters. That was really good and everyone's in time. So we've got time for questions. If anyone has a question, maybe just speak out, or if you wanted to continue your chat, you could also speak out with that.
Katie, there's quite a few questions in the chat as well.
Yeah. So from Jamie to Quinn, are you getting any reports of mortality events at the moment with the current heat wave conditions?
Oh, I think absolutely. Unfortunately, we're hearing reports of mortality and poor quality at offload at port already. It's quite concerning because it's really quite early in the season. So unfortunately, I think things are going to get worse. And I also think importantly is that the impacts aren't actually just during the heatwave events. They're much longer lasting. you're seeing flow-on effects into following seasons. I think those flow-on effects are mainly through disruption of molting cycles. So molting lobsters are getting landed, but there also can be some potentially prolonged physiological stress associated with it. Yeah, both here and in New Zealand, we saw impacts for several seasons past the heat wave, which was a bit of concern.
Right. So those improvements in post-harvest will get us like so far, but we're still expecting.
Absolutely, we've got to do what we can. It's clear there's improvement that can be made, and it's clear that what may have worked in the past doesn't necessarily work now, and so it's a matter of just everyone trying to do their best, and everyone realizing that there's a lot of hands involved getting a lobster from the ocean to China. There's a lot of processes, and so everyone needs to do their part.
Katie, it's Em here. Just looking through the chat, there's a lot of interest in not only the immediate effects on mortality this season from the heat wave, but both for rock lobster and abalone, these effects on longer term recruitment. And so one of the questions in the chat, to go back to Quinn, was whether there was any anticipation of a knockdown in recruitment because of this heat wave that might be seen in later years.
Look, absolutely. Anything that causes physiological stress is going to have impacts on key, physiological process being growth and reproduction. And so it would make sense that, if the animals are going to get stressed, they're going to have less energy and less prioritise reproduction. So you would think that there could be a longer term impact on that. Lobster recruitment's very complex with their long larval life cycle. And so, trying to quantify those would be very, very difficult.
Thanks, Quinn. And so, Jamie, I noticed you answered a question in the chat about the effects and the differences possibly between black lip and green lip abalone. And in your answer, you identified that there really are some important spatial differences too. And so my question is that going to be the emerging research need and assessment need we've got now to do this work at the relative fine scales to be able to better understand these relationships?
Yeah, well, certainly from some of the preliminary work we've done through the collections of blacklip along the east coast, there's definitely a spatial, spatial adaptation that we're seeing in these physiological processes. And if you think back to the 2016 marine heatwave event, yes, we saw some instantaneous mortality and we lost potentially 5 to 10% of the stock, but I think it would have been a far more catastrophic event, I think, and we would have seen a lot more mortality if there wasn't some localized adaptations occurring. So there's obviously some genetics at play here in some of these localized populations and some adaptation. And naturally, we seem to think that, looking at some of the preliminary work, that some of the northern and eastern populations potentially have some localized adaptations. And a lot of the sort of mortality that we did see back in 2016 was more in the sort of lower east and east coasts. So yes, I think there is some spatial resolution to this, and certainly what's coming through in the data at the moment is reflecting that.
Cool. That kind of leads me to a question I had for Daniel about if wild fish can either, extend their range or change their range or evolve a new thermal optimum. What can you do about this in aquaculture? Is there a way for them to be evolved?
Yeah, good question. So some species do have the ability to adapt, just to a certain degree, to thermal stresses or low oxygen concentrations, but more importantly, a genetic variation that exists, within individuals and within populations, and it's really about understanding those genetics and using those genetics to then use for selective breeding programs moving forward, so that they're more tolerant to these adverse conditions.
Okay, and then going back to the chat, we've got one from Jamie, one from Zoe to do with biotoxin monitoring and the Palawa population that use wild fisheries. And is there a way to help, from Jamie, to help identify toxins and harmful contamination when wild harvesting?
Yeah, so I guess the tricky thing with biotoxins is there's no apparent signal. Like we can't tell for it by just looking at the seafood whether there's any toxins in it or not. So it requires chemical testing. And while there are rapid test kits that are being used on some of the shellfish farms, it's a bit more involved. So there's no ready way of telling. But the best way would be just to jump on that public health website and see if there's any biotoxin activity detected, and then just looking which areas you harvest in.
Yes, okay. And leading from that, is there biotoxin monitoring in the Furno, from Zoe? Question.
Yeah, there's one monitoring site up there.
Okay. How expensive are those test kits? Like, what would they ever be available to the public?
Potentially. So we've spent a lot of time, like over 10 years, looking at a rapid test kit and validating its use and making sure how well it works in different seafood matrices. And they've just took the test kit off the market this year, so that was a bit of a blow. And we're looking for replacements at the moment. Hopefully down the track we'll have something that is a bit more of a visual one where we can see, you know, presence, absence, like a rat test for toxins essentially. But there's none available at the moment.
Yeah.
Katie, can I ask Andreas, how often do they do that testing for the biotoxins?
So it depends on the season. There's generally sort of higher risk seasons, especially on the East Coast, that are associated with Alexandrium catenella blooms. And it's generally every 2 weeks. And if there's biotoxin detected, it goes to weekly.
Anything else from the chat, Em?
I think we're covering most of it. I think, Andreas, are you happy for us to share your contact details so that people like Zoe and Jamie can get in touch with you just to understand more about these programs?
Yeah, absolutely. And I'll pop the Safefish link in the chat as well, just so there's heaps of information on that website.
Excellent.
Katie, I can see one question we haven't answered there about Andreas, with the coming marine heatwave, can we expect a higher risk of Vibrio, something I can't pronounce?
Oh, yeah.
Parahemolyticus. Yep.
Yeah, that thing.
Yeah, the simplest answer is, you know, if you think about temperature, increased temperatures, you get more microbial growth. But again, like we heard yesterday in some of those biological talks, it's dependent on the different species, different strains within that species. And not all Vibrios are harmful, so there's some that are more problematic than others. And the other thing we don't really understand at the moment is what makes them, so the virulence of them, how when are they actually expressing these things that are harmful to us? And that's ongoing work at the moment, trying to see what the risk is. And the best thing we can do is the post-harvest control, so chilling them down as quickly as we can and maintaining that cold chain.
Is there anyone else that wants to jump out? One for Jamie and or Quinn. Is there anything, like, we know this, another big heat wave's coming, and we know it's going to impact recruitment and molting and for lobster and recruitment for abalone, and just general mortalities. Is there anything we can do about it, like reseeding or translocations or anything like that?
I suppose I can answer first. Absolutely, in the southern rock lobster fishery, there is quite a large translocation program, which has, I think the evidence out there is having a really positive impact. We're also considering some on-growing of purulus into slightly larger, more robust lobsters to be released into the wild, and particularly in key areas such as the East Coast. And so, yeah, trying to help those populations and reduce that mortality that occurs early on in the post-purulus stages is something that we're definitely interested in.
Yeah, and I guess from an abalone perspective, it's probably something that we haven't we've tried to avoid in the last few years. There has been a lot of discussion around reseeding for other purposes, just to rebuild our fishery from the depleted state from overfishing, and it comes down to a lot of the biosecurity issues with abalone and some of that genetics and whatnot. So it's something that we haven't really looked too much into at this point in time. And I think one of the key things here is that, you know, if we think back to that 2016 marine heatwave event, if there wasn't some genetic diversity in the population currently, then I think we probably would have seen a far greater mortality event. So, you know, harvesting or removing these animals now, you're potentially going to sort of take out that genetic adaptation that's already existing there for some of the population. So yeah, it's a bit of a catch-22, I think, at the moment to start going down that route for abalone, and the cost benefit of it would be quite substantial, I would have thought.
Back to you, Em, for the final slide, or?
Thank you, Katie.
Yeah.
I shall reiterate again, thank you to all our presenters for their fantastic insights, and to Katie, particularly for chairing this session. So just a reminder, as it's been in the chat, all of the materials that have been generated and links to upcoming webinars, your point of truth for all of that is this CMS webpage. So we've got two more one on Thursday and one on Friday. Tomorrow we're really switching to the action focus, and we're going to be hearing from the Palawa community about their marine heat response plan in the Furno group, and also from the department about their marine response plan and the seafood industry Tasmania. And then on Friday, we'll be talking, Greta will be hosting a session on what are some strategies to recognise and address climate distress, which is really going to be something we're confronting across this summer. So thank you again to everybody for all your contributions and the interaction through the chats. It really gives us a signal of the information that's needed and what's valuable. And again, to our presenters and to Katie. So just because we always like to leave on a note of there being resources and places and means of having action, we just recall you again to this marine heatwave flyer that the department has circulated for actions this summer. And we look forward to continuing the discussion with you in the following days. So thanks again, everyone.